Welcome to Synexa Life Sciences. Synexa is a specialist provider of biomarker and pharmacogenetic services to the global biopharmaceutical industry. We offer analytical and advisory services to drug and diagnostic developers focused on the identification, development, analysis and interpretation of biomarkers in clinical trials and diagnostic validation programs. The company was founded in 2003 by its current management team and has grown rapidly as biomarkers have become an important decision-making tool in drug development. Today, we employ around 40 scientists operating from our custom-designed lab facilities here in Cape Town.
Synexa provides an end-to-end set of biomarker services, including the discovery of new biomarkers, the development and validation of biomarker assays, advice on selection of biomarkers for clinical trials that takes into account the optimum value, both economically and scientifically, of those biomarkers for the study in question. We support clients with a broad portfolio of biomarker assays, including genetic markers, soluble protein markers, cellular markers, and tissue markers. And lastly, we provide interpretation of results to help clients extract the maximum value in terms of safety, efficacy, and the identification of other applications for their drugs based upon the biomarker information at hand. At Synexa, we focus on building collaborative partnerships with our clients. We are not a typical central lab offering a simple menu of assays. Instead, we invest a lot of time up front with clients to understand exactly what they are trying to achieve, and we design a biomarker program to help generate new insights and improve the quality of decisions that can be made about their drug. Collaboration like this requires deep and creative thinking, a willingness to take on challenges and push the boundaries of what's possible to solve unusual problems. This entrepreneurial, open-minded approach is central to our culture and guides everything we do, from how we recruit scientists to how they perform their daily work.
Something we pride ourselves on at Synexa is our innovative use of the latest scientific research and technology platforms to solve problems for our clients. For example, we consider ourselves pioneers in the application of flow cytometry as a tool to provide proof of concept as far as the mechanism of action of the drug is concerned, looking at the action of the drug at the cellular level. We can determine the response of the cells to the drug in the test tube, in other words, the proof of concept stage, or applying the same technology to follow the response of the individuals during the clinical development phase. This allows us to determine the exact cellular target of the drug and how that drug influences cell function. As an example, we are looking here at a subject's cellular response to a potential immune modulatory drug. You can see on this dot plot, the individual's white blood cells have been stimulated to undergo an immune reaction which we are measuring in the form of the secretion of important immune factors. If we now pre-incubate the same individual cells in the presence of the immune modulator, we are measuring here an increased response of the same factor. This suggests that this drug has the ability to enhance that immune reaction in the test tube. Ultimately, this type of information can help the sponsor understand whether the drug is useful for the treatment of a primary immunodeficiency or simply can be applied to bolster an individual's immune response during a vaccine development. And therefore, the client can make a more informed decision about relevant disease categories for which this drug could be applied. A focus on innovation has also led us to develop new products of our own, such as a flow cytometry internal control called LymphoSure. This product we developed to overcome the poor shelf life and inconsistent scatter properties of existing products. This is the product that we can use to qualify the flow cytometry before applying clinical samples.
Synexa's laboratory platforms include the latest technology available for biomarker analysis, including in immunochemistry, ELISA, CBA multiplexing, mesoscale discovery, and immuno-PCR. In our cell-based assay labs, PBMC culture, whole blood culture, ELISpot, and P3 facilities. In our pharmacogenetics labs, RNA and DNA isolation, real-time PCR, array-based assays, sequencing and bioinformatics capabilities. In our flow cytometry lab, a range of flow cytometers from small point-of-care systems to large 8-color instruments. In our diagnostics labs, facilities for antibody arrays, gastrointestinal diagnostics, immune function diagnostics, reproductive immunology, and salivary hormone analysis. And in analytical chemistry, LC-MS-MS, GC-MS-MS, preparative mass spectrometry, and MALDI-TOF platforms. Synexa's technology platforms and skilled scientific team allow us to bring a very wide variety of cost-effective solutions to almost any client requirement. Our focus on collaborative partnerships, innovation, and cutting-edge technology guarantee our clients a world-class biomarker service. If you'd like to learn more about what we can do for you, please visit our website where you will find our contact details.